Thank you, Lisa, and thanks for all coming out on a hot afternoon. It'd be nice to be outside than inside. Loud. Stand straight. So one thing that Lisa missed out on, I'll explain in a minute how I came to be working on both ticks and wildlife. But just by way of what this talk is going to be, really what I wanted to do is just thank everybody that I've worked with and put a lot of work into projects that I've kind of been involved in. And I thought the best way to thank people was actually to show the rest of you what the outcome of all that hard work is. So I'm going to do this whirlwind tour through a whole series of different projects. And there's not going to be enough information on any of them. But really the point is just to name check the collaborators along the way and to say thank them very much for all those hours of effort. So just to fill in that one little bit that Lisa missed. It's not going to work. All right, I'm going to hit. That'll work. The bit she missed was that the first research I had did was on cockroach physiology at University of Canterbury. And so I had this kind of misspent youth as an insect physiologist. But I then, the first job that I had was with the, I guess I'll come over here, was with the New Zealand Forest Service. And that was working on deer, and the deer over there are quite big. The red deer introduced from the United Kingdom. And they're considered a noxious pest, and so we do things like fly around in helicopters and shoot them with semi-automatic rifles. It's a very different system than here, and the kind of work I did was wildlife damage management. But while I was with the Forest Service, I was given the opportunity to do a PhD in Canada, in Alberta, and that work was on bushy-tailed wood rats. And so I'm going to show you a slide that's embarrassing, but I'm showing you this for two reasons. Firstly, to demonstrate that you always start looking like you study animal. But secondly, this was my first encounter with ticks, because that hair was curly enough and bushy enough that I didn't always notice when I had a tick attached. And there was this one weekend where I asked one of the other grad students out on a date, and we were at this nice candlelit restaurant, and we were kind of chatting, and I brushed my head, and a large engorged dog tick fell on the table and started walking towards her. So I won't go beyond that. Moving ahead, 25 or something years later, I've met my wife, Diane, who's actually watching the seminar from New Zealand. She was an American. I had done a number of projects to do with bovine TB, deer, possums, things like that. We moved to the States. We ended up at Michigan State University. And at that time, 2004, there had been a surge in Lyme disease. So a tick-borne disease, mostly up in the northeast and upper Midwest. At that time, there were about 20,000 cases a year. It's now almost double that, just staying. And as you can see from that map, it was very geographically patchy. It was primarily in the sort of Wisconsin, Illinois, and then out on the East Coast. And as a super quick background, what is Lyme disease? Because I'll talk about it quite a bit in this talk. It's a tick-borne disease, and the vector is the immature life stage of the blacklegged tick, which I'll show you a picture of. And it's a bacteria. It's a spiral bacteria, a spirochete. It literally bores through connective tissue. When it infects you, ends up in lots of different organ systems. If it's untreated, it can go from being like a rash to being neurological disease, heart disease. Lots of bad things can happen if you don't treat it. So it's not good that there are something like 20,000 cases a year. And that tick that I mentioned, here's pictures of the male and female of 3 different tick species. Black-legged tick, which we also call the dog tick, lone star ticks in the middle, and, sorry, did I say dog tick? I mean deer tick. Deer tick, lone star tick, dog tick. And anytime I'm talking about Lyme disease in this talk, I'm just talking about this species. These ones do not spread Lyme disease. Now that's a controversial thing to say, particularly down in the south, But one of the reasons I can say that is that, and this is my first call out to a collaborator, I was involved with Ellen Stromdow from the Army Public Health Command, and she recently did like this massive review, as it says there, 60 publications, tens of thousands of ticks tested, basically arguing the case that the Lone Star tick is not a vector of Lyme disease. And that's the main tick down here in Tennessee. So we're going to come back to that later and I'll tell you horror stories about it. But for now, with Lyme disease, we're not talking about that guy. So 2004 up in Michigan State, as I had kind of arrived, it coincided with Centers for Disease Control wanting to get a better handle on that spatial map that I showed you. That map was based on human cases, but humans move around. Human case data is difficult to interpret geographically because people drive their cars from state to state. And so CDC wanted to get a risk map based on where the vector tick was and where that tick was infected. So abundance and prevalence of pathogen. And we, Michigan State, became involved with the consortium of several universities, Yale University, University of Illinois. And in particular, I hooked up with these folk, Dr. Jean Tsao, who had done her PhD at Yale, had recently come to Michigan State, and our first co-advised PhD student was Sarah Hamer, who's now at Texas AMU. And so the three of us set out to contribute to this group project on mapping the distribution of ticks. The methods were basically drag sampling, dragging a cloth through the understory. Ticks get on that, they mistake it for a host. Collect the ticks, you do a PCR, you look for the Borrelia bacteria directly. So you're not dealing with complicated human serology, you're looking at the direct DNA of the bug. And this project was large scale. It was essentially in the eastern half of the US from the bottom to the top. And it spanned over a couple of years, multiple sampling, lots and lots of undergrad students hired in their summers to do the field work. And as I said, I'm going to be skipping super fast through these things, but just as an example of the kind of data that generated, those circles are where we found nymphal ticks on the drag cloths that were infected with Borrelia. And you can see the redder and yellower parts are up in Illinois, Wisconsin, they're out on the coast. It matches very closely that human map of disease. But one thing that we did see from that was that there seemed to be a kind of a spread process coming down out of Illinois into Indiana, just kind of on the edge, southwestern corner of Michigan. And so Sarah Hamer's PhD thesis was asking what's going on in the lower peninsula of Michigan. She essentially ran transects up the coast and inland. And over the space of a 4 or 5 year PhD study, was able to demonstrate Lyme disease essentially marching across the landscape. It was very eye-opening for all of us how quickly that geographic spread was happening. And we think part of that is because it's driven by immature ticks being spread by birds. Now, while that PhD was going on, I pulled up stakes and moved to Fisheries and Wildlife here, Forestry, Fisheries and Wildlife here at UT. My wife moved to the business college across the river, across the railway tracks. And so, you can see from that map, the sampling, although we had done a lot of the eastern US, Tennessee essentially got missed. And so I arrived from Michigan to a state where there really was not a lot of concrete information about Lyme disease. And the very first conversations I had when I got here, when I told people what I had been doing in Michigan, was half of the people said, my aunt, my uncle, and my second cousin all have Lyme disease, and they got it in, Rowan County or wherever. And then the other half, particularly the doctors that I talked to, said there are no black-legged ticks in Tennessee. It's impossible for people to get Lyme here. And so one of the things that we had been doing up in Michigan was going out in the fall to deer check stations. The adult ticks of the species are active in winter. They love to feed on deer. Deer are kind of like a cruise liner. You kind of get a meal and get to mate. And so that's where you find the adult ticks. And so the very first fieldwork that I did was with Dr. Reed Gerhard, Dave Paulson, who I don't think is here, but he and I have done many projects since this. Reed retired, and also Robin Sissel, who was a PhD student over in the vet college at that time. And so it was like the most simply answered question ever. We drove out one night or one morning to Oak Ridge. We looked at a handful of deer. We pulled ticks off them. There were lots of ticks on the deer. Most of them were kind of brown, so that was not black-legged ticks. But we found about 6% of the ticks were black-legged ticks. So as I said, it was like the easiest question ever to answer. There are black-legged ticks, at least in the first place we looked. And if you looked in terms of the infestation on the deer, about 15% of the deer in Anderson County at that point were carrying black-legged ticks. And so that initial data point motivated me to take on Michelle Rosen as a master's student. Michelle's project was to expand that right across the state. And so if you can see the little yellow dots there, she worked with the fish and wildlife clubs at all of the Tennessee universities, recruited folks to go out and do exactly what I showed you before, go out on the weekends, work with TWRA, pull ticks off the deer, send them into Michelle. Michelle did a species determination and then tested them for Borrelia. And so the kind of results she generated, the red circles there indicate what percentage of the deer were infested with the ticks. The green in the background is kind of a measure of how much sampling we did. So we didn't do every county in that survey. But you can see that, black-legged ticks, most deer in Middle Tennessee were carrying black-legged ticks back then. However, out to the west and out to the east, a lot of them weren't. And in fact, we didn't get any black-legged ticks along that kind of eastern range of the Smokies. And we will come back to that a decade later, a little bit later in the talk. So we've got lots of black-legged ticks, some geographic pattern. What about the, that was just saying, Middle Tennessee with lots and the foothills without. And then what about the Borrelia? So we had the ticks that came off the deer. We also supplemented that in several subsequent years with collecting flat ticks, unengorged ticks from the vegetation using the drag cloth technique. And those came from several different states, several different counties. And when those were tested by PCR, we didn't get a single one positive with Borrelia. And that's unusual. You're up in the Northeast, if a tick population is infected, you would expect 20, 30, 40, maybe even 50% prevalence. It's not a hard disease to find, or a hard pathogen to find. So back then, we kind of concluded that yes, black-legged ticks are widespread, but they don't seem to be carrying the Lyme disease pathogen. And so our conclusion at the time was black-legged ticks and Lyme disease are not the #1 thing that folks in Tennessee should be worried about if they're worried about ticks. And that in part made sense because black-legged ticks aren't the tick that usually bites people. This pie graph is based on, it's actually quite a long span of years, but folk out at Oak Ridge and I blanked on the name, but the folks that kind of monitor the environmental quality out there, they send any ticks that end up on their field staff in each year, and just aggregating all that data, what you see is that the tick that commonly bites people in the field here is the Lone Star tick, and then second on the list would be the dog tick. The gray are the black-legged ticks that we've been talking about. There's a few of those. The yellow are Gulf Coast ticks, another amblyommii species that are starting to sneak into eastern Tennessee. So I'm going to change tack and talk about the Lone Star ticks for a bit. And that was essentially what we did with the research program. Having kind of answered the question of black-legged ticks, we thought, we shifted over to think about amblyomma for a while. So what happens when amblyomma bites you? Well, I think many of us have probably had that kind of a rash. You get a bite, you get like a small, red, itchy rash. It can last quite a long time. The ones that last a long time, we've probably had some sort of bacterial injection. There's a thing called Rickettsia amblyommii that's in many of these ticks. Back 5 years ago or more, I would have said it's not pathogenic, it has no zoonotic potential. We've changed our mind on that. We now know that it can be associated with some fevers. There's probably different strains of it, and some are more virulent than others. So that's one of the things that you can have happen. You can also get a rash that looks exactly like a classic Lyme disease bullseye, but it's coming from a Lone Star tick. It does not appear to be bacterial. It does not appear to respond to antibiotics. If you don't treat it, it goes away again. I think it's kind of in the allergy world rather than the infection world. But the problem is, it's in some cases, it can be almost impossible to distinguish clinically for a doctor that and Lyme. And so because you can get that kind of rash here in Tennessee, I think that's one of the reasons a lot of people will think they've had Lyme disease. And some of the doctors may be confused about that as well. So there's that, but like I say, you don't treat it and there aren't bad outcomes. The real concern with Lone Star ticks is they do carry Ehrlichiosis, which is something that a few decades ago, human doctors really didn't have on their radar. It was a kind of a livestock and a domestic canine disease, but not really a human disease. Tennessee has the claim to fame of having, in the early 90s, the biggest outbreak of human Ehrlichiosis the US had seen, big enough to the extent that CDC did an outbreak investigation. And this was up on the Cumberland Plateau in a golf-oriented retirement community. And what had happened was essentially developers had gone in and bulldozed down forest, put in fairways and greens in those bulldozed areas, built houses around the edge, and then filled it full of somewhat elderly, somewhat immune-suppressed clients. And so when CDC did their outbreak investigation, which is published, and I had actually heard about this study in New Zealand before I came here. They did a classic case control study, and the two risk factors that popped out were firstly, the presence of ticks, fair enough. The other one was golf handicap. Now, why do you think that was? What would happen if you had a high golf handicap? You hit the ball in the woods, you go in after the ball, and you get bit by ticks. And the doctor that I knew in New Zealand had heard of the study because he was like, this is awesome. I can prescribe that you need to improve your golf game. So that had all happened before we arrived. We got involved because the community managers there were trying to do something about the problem. And the method they were trying was what's called a four-poster. It's like a big hopper of grain that attracts deer. They feed out of the grain here, and these paint rollers wick down into a reservoir with the caricide, and the deer brush themselves against that. They do a topical treatment of caricide, and it gets rid of the ticks. And all of that works as advertised. The deer do come, they do treat themselves. But the question was, is that reducing the tick population? Is it having any effect on pathogen prevalence? And so my second master's student, Jessica Harmon, came in to look at that question. This was a map of the golf-oriented research retirement community. They had 8 feeders up at the red dots. And what we did was we watched what was happening at the feeders using trail cams. And so in this case, middle of the day, middle of summer, deer got kind of tired or lazy and like lay down in front. And that's kind of neat because typically in midsummer, you might expect to see large numbers of black-legged ticks. Sorry, Lone Star ticks infesting a deer at that time of year. And he's clean, bushy, clean as a whistle. So deer came, we saw lots of deer, we saw lots of other species as well, which is a little bit of an issue that you're kind of feeding every wild animal in the woods, possums and squirrels and raccoons. We can talk about that separately. But did it reduce the tick population? Well, it did, but it was a short range effect. So this goes out to about 300 meters from the feeder. That was about the scale of the effect that we could measure. And if you put a 300-meter buffer around those feeders, you can see you're really not treating a significant part of the area. And so they kind of work, but on a scale that is too small for an area that size and is too small to be affordable. I mean, this program was costing the community a lot of money as it was, and it wasn't given proper coverage. And unsurprisingly, when we tested the ticks for pathogens, we found 3 different species of Ehrlichia. All 3 of those are zoonotic, not super common, fortunately. They're in the kind of the 1 to 2 to 4%. But when we compared that in this treated 4-poster area with an adjacent area, there was no difference in prevalence. And so that was my one attempt in the 14 years to actually fix the tick problem. And then I kind of gave up and said it's too hard and went back to just watching the tick problem. And so as an aside, when people say, what should you do? We very much push personal protection. Tucking your socks in, being a fashion don't, doing a tick check at the end of the day, long pants and things like that. And so I've used that slide quite a lot. And I went to Jessica's final defense, and she used the slide too, except she modified it slightly. I will get my revenge one day. Now, skipping a decade ahead, but just while we're on the topic of Lone Star ticks, the other bad thing that can happen with Lone Stars is that they are the one that spreads red meat allergy. It seems to be becoming a lot more common. I'm not sure if it's partly that people just now have a name to it, but the deal here is that it's not an infection, it's an allergy. It's triggered by the saliva of Lone Star tick bites, and it creates an allergy against a particular sugar that's associated with mammalian meat. Actually, not all mammals, not old world primates. So you could still be cannibals if you wanted, but any of the other meats, and it can also sometimes generalize to milk and things like that. The particular sugar is called alpha-galactose, so they nickname it an alpha-gal allergy. The really distinctive thing about it, it's unique from pretty much all other food allergies. Well, two things. Adults can develop it, and allergists originally were very resistant to the idea of how could you develop an allergy to something you've been exposed to all your life. But the reason is that the tick is injecting it directly into your blood, whereas normally you're exposed to it through the gut. And that seems to trigger the issue. The other thing that's unique and diagnostic is this 3 to 8 hour delay. So basically, if you have a burger at dinner time, just as you're going to bed is when you start feeling sick. And for some people, this is very, very dramatic. Diarrhea, vomiting. They will recommend that people carry an EpiPen if they develop this allergy, and it can last literally for years. It often will gradually fade, but it doesn't fade in a week. Fades in months or potentially years. And so I don't think Janetta will mind me saying, unfortunately, Janetta developed this during her master's project, and I mean, I think you're still way high antibody titers. So how often do you get checked for that? Yeah. So there is an antibody test. You can go to Med Center and they will say, this is the level your antibodies are at, and come back in a year and we'll see if they've gone down. So a number of talks recently when I've come to talk about things like Lyme disease and Ehrlichia, people are like, whatever, we're done with that, tell us about meat allergy. It's really developing a high profile now. Okay, how am I going for time? Okay. So, the other thing I'll say is there's quite an interesting National Public Radio Lab show on alpha-gal. One of their producers got sick. I was the tick guy they interviewed. So that was actually one of the highlights of my 15 years, being interviewed by NPR, sat in the studio over here. And the one problem was, like, they were really fun to talk to, and we talked for at least an hour, and I had to get kicked out of the studio because we'd gone over time. And so I didn't get to read the credits at the end. You know how they do that? If you listen to that show, they get all the people who've interviewed to like try and pronounce their names. So everybody that heard me on this, and this thing, like people have heard it in New Zealand, it's gone really widespread. People always ask me, how come you didn't read the credits? It's not like they paid any attention to what I said. Oh well. Okay, so now, after an interlude on Lone Star ticks, 2010, we had had several goes at getting NSF funding to work on this kind of geographic pattern of Lyme disease, and that was all being done with the the collaborators up at Michigan State, and actually with a number of other universities. And so in 2010, we finally got one of the NSF Emerging Infectious Disease Awards. And so there was a cast of it was a multi-institution award. I won't go through all the details there. But the question was what I've been kind of hinting at all the way through this talk is, why do we have this distribution of the human cases up in the northeast and midwest when the distribution of the tick itself, we're back to black-legged ticks, is much more widespread. So that was the motivation for like a 4-year NSF grant. And I'm actually not going to talk about any of the projects that were the big consortium projects, because I don't want to like claim I was just one cog in the wheel of those. What I'm going to talk about for the rest of the talk are the bits that we kind of did on the side with our grad students, our REU students. And the first of those was just a very small specific question. Well, the bigger question was that one of the possible reasons is that although there were black-legged ticks in the south, maybe they just weren't abundant enough. Densities weren't high enough to maintain disease cycles. And one idea we had early on was, well, maybe it's fire ants. Fire ants were rampaging and invading and eating things. Maybe they hammer the black-legged ticks. And so the first project I'll tell you about very briefly was an REU student, Patrick Minari. We worked down at Tall Timbers Research Station looking at, in various ways, interactions between fire ants and black-legged ticks. In this particular study, we were like literally putting out little engorged tick nymphs near a fire ant mound. And when you did that, you definitely saw an interaction. They picked them up and immediately ran away with them. And so one of the findings was, an engorged tick full of host blood is probably a tasty meal for fire ants and other predatory ants. The thing down at Tall Timbers, though, was there were plenty of ticks and there were plenty of ants. And on a microhabitat scale, they lived in different places. Fire ants tended to be in the driest areas. Black-legged ticks tended to be down in damp gullies, and they just were able to coexist in that way. That was just an REU project. I think there's actually more to be done on this whole question, but we did not ourselves do more. But we did go back to that question about what are the densities of black-legged ticks in the south? And I used to go with my wife and my son, who, by the way, is in the audience, Jamie, hi. We would go down to Jacksonville. And while Jamie's like surfing in the surf and doing whatever, I would like wander along the side of the beach. And I'd be going like, man, there are a heap of black-legged ticks here. So that was like, 20 minutes walking along with, I'm not even sure if I had a drag cloth. And those are all, they are the right species. They're not, as far as I know, infected with Borrelia, but they're there and they're common. And so one of the projects that Lauren Meisters, my next master's student, did was basically do a survey transect, dragging ticks from Virginia all the way down to Florida. And the results that he got kind of reinforced that it's not just an absence of ticks that's the issue in the south. He actually got a significant increase in abundance as you went south on the coast. So the bigger the circle, the more ticks per hour we were able to drag. Now, I do have to say that pattern isn't what you see inland. This is a coastal issue. And so if you look at, obviously the question is, are they carrying Borrelia? When you looked at that, you got an even stronger geographic pattern, but in the opposite direction. So the Borrelia was prevalent up in the north, and then as you came south, essentially dropped out of the tick population. And so we answered the question, no, it's not abundance that's the reason we don't have infection down south. Now that pattern, people kind of knew that there were ticks without infection in the south, and one of the arguments that have been made is called zoo prophylaxis. Essentially what that was that in the south, the hypothesis went that the immature black-legged ticks like to feed on lizards. So that had been published initially by Charles Epperson from North Carolina. There were several other studies along those lines, but then there had been some studies kind of arguing back. And the argument was about whether the lizards could maintain the bacteria. Were they a reservoir competent host? If a tick came and bit a lizard, would it become infected? If another tick came along, could it pick up the infection from it? And so the next project we're involved with was Teresa Moody. She came in to answer those two questions. Can you infect a lizard, and can a lizard infect back to ticks? She collected wild ticks in southern states. She set up a lab study over at JAR2. She had as a control mice, because we knew that mice are reservoir competent for Borrelia. She put infected nymphs on them. She tested those nymphs after they'd fed on the lizards, and then later she put some larvae on the lizards and uninfected larvae to see if they could pick up infection. So here's the results. She put on nymphs. About 75% of those nymphs we knew were infected, and they fed on the lizard, and then they dropped off. And when she tested them, a few days later, once they had dropped off, most of them had lost the infection. So lizard blood is actually a kind of a treatment against Lyme or against Borrelia. And then, meanwhile, over in the controls, like all the mice became, all the nymphs that fed on mice retained their infection. So mice were fine, lizards zapped the bacteria. Then she put on uninfected larvae, let them feed, let them drop off, tested them, none of them picked up infection. Whereas with the mice, all of the larvae picked up infection. So mice are a very good reservoir that will kind of keep the disease transmission going. Lizards break the disease transmission cycle. So that, in a lab study, reinforced the hypothesis that these previous field folk like Apperson had proposed. Then we wondered, okay, maybe that is something that's only associated with southern ticks. Maybe the northern ticks don't like to feed on lizards. Maybe if northern ticks came south, I'll get into that. We had some reasons to think northern ticks might be moving south. Maybe, they would still continue to feed on mice. And so it's an REU project, also down in Florida. REU student Meghan Fitzgerald worked on that question. And so what she did was she set up some enclosures, and in those enclosures she put tick nymphs, some from the north, from Maryland, some from Tennessee, and some from Georgia. So each of these tanks had 50 nymphs sitting up there kind of waiting to grab something. And then we applied one mouse and one lizard to each of those tanks. We did this repeatedly, so there was replication to get the stats. And what we saw was, if we just look at the Maryland northern nymphs, blue as mice. So, if you went on the mice and if you went on the lizards, no significant difference. They essentially didn't care. If there was a host there and it came by, they would get on. But that wasn't what we saw with the southern nymphs. Southern nymphs had a strong preference for getting on a lizard rather than a mouse. That was statistically significant. And so this was one of our first indications that there are these behavioral differences between northern and southern ticks, even though they are all the same species. And if you take a Maryland tick and a Georgia tick and put them together, they will mate and they will produce viable offspring. But nevertheless, there are these geographic differences in behavior. So while all of this was going on, it was all very kind of nice and theoretical, things were kind of coming unstuck on the public health front. So from 2000 through to 2010, there was a massive surge of human Lyme disease, and this was all being measured by the health department, coming down the eastern side of the Appalachians. And so by about 2010, we had this hot spot of human infection down in Southwest Virginia. And I'll show you, here's a health department map. This is even more recent. This is kind of just several years ago. But that red area is a prevalence of more than 100 Lyme disease cases annually per 100,000. That's equivalent to the worst areas up in the Northeast. That's crazy. And to be honest, I didn't really believe it when I first saw that. I thought this can't be right. Because it's only about 2.5 hours drive from Knoxville. And we had done all that survey work saying we don't have any infected ticks. And I'm going, I don't know about that. And so literally one weekend about 4 years ago, I drove up to, this is the Virginia Creeper Trail, the New River Valley, that kind of area. And I went to a kid's playground that somebody had said they had seen lots of ticks at, sampled ticks, brought them back to the lab. Half of them were infected with Borrelia. And I'm like, whoa, that's kind of scary. And I told Christy earlier I was going to like scare her at some point. So this is where the scary bit starts. Because that obviously raises the question of whether that arrow that we saw coming down the eastern side of the Appalachians, is that going to continue on into Tennessee? Because it seemed to be kind of tracking the valley system. And so crossing over into Bristol, you've then got, coming down into the upper Tennessee Valley. And so the next master student was Janetta Kelly, who's in the audience. And she was from Kentucky, but she was doing her undergrad here. And I said, Janetta, we need to figure out like what's happening in Tennessee and in Kentucky too. And I don't know that they would understand my accent in Kentucky, so you could help. I'm exaggerating a bit. I more had that problem in Alabama. Alabama, I couldn't understand them, they couldn't understand me. I can understand folk in Kentucky. And so what Janetta and assistants and myself did was essentially rampage around the countryside in the fall of 2017 and 18, dragging with our drag cloths, seeing if we could get black-legged ticks, and then testing for Borrelia. And remember, this is what we had done 10 years earlier, and we had never got any positives. This time around, we did get positives. So out on that side, those red dots are where we got black-legged ticks infected with Borrelia burgdorferi. So out in Virginia, in the New River Valley, it was just like I'd seen in the playground. Everywhere we went, about half the ticks were infected. But we also were getting positives up here. Knoxville is about here, I think. So this is kind of Oak Ridge, places like that, coming up into southeastern Kentucky. And one right down, this is Hamilton County. This is just on the outskirts of Chattanooga. But all very much associated with kind of the river valley systems. And so those were the first confirmed Borrelia in eastern Tennessee. We've got a emerging infectious disease paper out of that. That work is very much in progress, but I'll just show you a couple of slides to think about. That's Chuck Swan Wildlife Management Area up in Union County. More than half the ticks there are infected with Borrelia. And that's, the other side of the lake is marinas and kids paddling and mountain bike trails. So that's kind of worrying. I had a phone call, or actually through some of the entomology folk, about Concord Park out in Farragut. A lady that jogged out there with her dog said, my dog keeps getting these ticks on them. And when we looked at them, they were black-legged ticks. And when we went and dragged, that's like a couple of hours dragging. Jamie and I went out there one afternoon in last November. There's a heap of black-legged ticks out there, but none are infected. And so somewhere between Union County and Knox County is this kind of threshold. And the scary thing, Christy, is that may well be moving south and west. And so that's something that needs to be tracked. The other quick comment I want to make about Janetta's work from her survey work is when we found these positive ticks, you can genetically type the bacteria and look at kind of relatedness. There are various restriction strain types. And so we were doing that to see if we could get a clue of where Borrelia in Tennessee was coming from. And one of the strain types, which is associated with the Northeast, did exactly what we had kind of hypothesized, that it was linked up to that Southwest Virginia Lyme hotspot. And so we're seeing that down in the counties just around us. But there was also one that was kind of associated with both Virginia and Kentucky, and then was down in Tennessee. And there was one that was only in Tennessee. So there was really quite a diversity of strains. It kind of was not what you would expect if this was like a super recent invasion and they had all come pouring across the border from Virginia. looks like there may be some invasion coming down the western side of the Appalachians as well. There may be some long-term Borrelia strains, maybe associated with the high deer numbers and ticks we saw a decade ago further west. So lots of questions there. And I've said to Lisa, I think maybe I need to come back each year in the fall and learn more. Okay, so then the final thing I want to talk about, I hinted about these behavioral differences associated with feeding on lizards or mice. There's an even more fundamental behavioral difference, which is that, remember I said it was the immature ticks that create the risk. And the reason I say that is Lyme disease is a disease of summer. The adult ticks aren't the ones that are active in summer, it's the immature ticks. And they're small and they're hard to see and they're sneaky. And the thing is, down in Tennessee, even in areas where we could get lots of the adults, like Concord Park in November, it's very hard to drag the nymphs. But the adults are there, so the nymphs must be there. It's just they don't get on a drag cloth, and that's the behavioral thing. They're not on the drag cloths because they're not coming up out of the leaf litter. They're staying buried. It could be like desiccation or heat, or maybe that's where the lizards are, or whatever. This behavioral difference. And so we wanted to look at, what's the geographic pattern of that? And so PhD student based at Michigan State, but I co-advised them, did what we call the tick garden study, which was to bring in ticks of different genetic origins from the northeast, the northwest, the south, put them in a common garden. We did this work both in Michigan and then here, and by here I mean out at the FREC with Keith's collaboration, which we really appreciate. And so a tick garden was just these circles, arenas made of aluminium flashing with a frame on to stop deer and raccoons messing with it. And inside those we had these little dowels. We would add nymphs, some would be southern, some would be northern, depending on the experiment, and we would simply score whether we ever saw them climbing up on those dowels. We did that daytime and nighttime. And when you did that, you get graphs like this. This axis is what proportion of the nymphs did you see climbing, and on that axis, that's kind of ranked by latitude. So the red are Lyme endemic areas of the northeast and upper midwest, places like Michigan, Wisconsin, Minnesota. The green are these areas that have not traditionally had any Lyme disease. And you can see that there's, in a common garden situation, same environment, same rearing, there's this genetic difference in that propensity to come up out of the leaves. And that's the thing that puts humans at risk, coming up out of the leaves and like biting your ankles or climbing up and biting on the butt or whatever. So the interesting thing there is Tennessee is very much intermediate. And so there was a couple of possibilities. One is that Tennessee is a mixture of northern and southern ticks. So maybe there's been southern ticks here all along, and now the northern ones are invading. It's also possible that they're genetic hybrids, so that what I just said applies, but also they've mated with each other. And so now you've got these ticks with intermediate behavior. And so if I had had the opportunity to stay longer, that was the thing I was going to work on next. And so I've got to lob the ball to someone else to do that. But a genetic study of whether we've got hybridization going on, and also whether that red boundary is going to move south. Okay, I'm not going to keep you guys from the food too much longer. That's the paper that came from the behavior study I just talked about. So that's a 2019 publication, which I'm kind of proud of, and I think is a good note to end on. I guess to have some conclusions, black-legged ticks are now well established in the Tennessee Valley, and we've got several counties, the yellow ones, that we know we have infected populations, and actually can add Johnson County to that since this map was drawn. We see those foci of infection associated with the big rivers and lakes, and I think that's probably a humidity thing. Tennessee is basically a bit hot for these ticks, but if they hunker down near a lake or a river where the humidity is higher, they can do well. People always ask me, what will climate change mean for ticks? I think everybody's always thinking about it getting hotter. Getting hotter would actually not be so great for these guys. But one of the other predictions of climate change is that the southeast will get wetter and more humid. And I think that may already be having an effect, and maybe partly why we're seeing the southward spread of the northern populations. That may well continue. Tennessee Valley, therefore, I would say is probably at risk of seeing a massive surge in Lyme disease the way they saw in Virginia a decade ago. But having said that, what Virginia has is classic summer Lyme disease based on the nymphs. And I've got that photo up there just from a public outreach point of view. People have the wrong search image for these ticks because every photo in the media is the adult, which is big, the female's red. No. They are sneaky little tiny fuckers. And they are difficult to see, and they will get in nooks and crannies and be hard to find. So that's what you're looking for in summer. We're not seeing human Lyme here yet. I think presently, because of this behavior of the nymphs, the primary risk is in winter from the adults. And the thing in winter is, you know, you've got more clothes on. You're wearing boots, you're wearing long pants and so on. But you know who isn't wearing long pants and boots are the dogs. And so my kind of final point is, and we've talked to the vets about this, dogs are probably a good sentinel for emerging human risk because they will be picking up infection in these areas that we've been finding. And we do see that with some of the serological testing that gets done on dogs, that is starting to climb in counties like Anderson County and Union County. So I'm going to wind it up there. I've got some acknowledgements. Firstly, Kathy Scott was our lab manager and was involved in pretty much all of the laboratory assays in the early part of that talk. So she is very much sort of in the background as a key player. Rick Gerhold was the the postdoc for the NSF project. And I didn't talk about the NSF project parts, but he was very instrumental in setting up all the field areas in multi-state comparisons. So I'm very grateful to both of them. I'm obviously grateful to funding that came from AgResearch, Michigan State contributed, Tennessee Department of Health, and particularly Dr. Abelardo Moncayo contributed, and then obviously a big injection of funds from the NSF EID program. And then there are a whole lot of people that I did not specifically name check. For example, in purple up there is Emily Doob, who was Janetta's field assistant, and she was like fantastic to work with. All of these folk were, you know, assistants or other grad students on the project, and they were just like a fantastic crew to spend time with. I appreciate all of them. So I will leave it there with a photo of the New River taken from the Virginia side of the Virginia-North Carolina border. These woods are seething with ticks. North Carolina is worried, and I think should be worried, that those ticks are heading south on the other side of the Smokies as well as into Tennessee. So I will stop there. I don't know if anyone wants to ask questions because it's food, but I'm happy to take questions if anybody has any.
I have a first question. The tick that fell out of your hair?
Yeah.
Did you get a second date?
I actually did not. But I can't, to be fair, I can't necessarily blame the tick. We'll never know.
Second question, do you think we should be looking at local county populations for Borrelia since there's not so much kind of a better idea of
Well, I think,
that's our Lyme disease.
so in terms of the ticks or in terms of the Borrelia or both? I mean, yes, I guess I'd qualify that by saying it's a lot easier to trap a mouse than a coyote. But if you were working with wildlife damage mitigation folk that were like whacking coyotes anyway, they I mean, their advantage is they would attract adult ticks, but they would also be reservoir competent. One of the issues with the deer, I mean, Rick knows this, but for the rest of you, one of the issues with the deer is deer do not actually support the bacteria very well either. They're kind of lizard-like in that sense. So you always worry that if the ticks actually fed on deer, it may have a reduced detectable prevalence. Coyotes would be good from that point of view because the ticks would remain infected. So I think really it just comes down to if you have a ready source of coyote samples, it would be great. Okay, well, thank you all very much for coming.